For second part of our lecture, the cell-mediated immunity. CMI or cell-mediated immunity consists of T cells. T coming from the word thymus, where the lymphocytes from bone marrow migrate to thymus to become immunocompetent. So the T coming from thymus. In the thymus, the immature T cells, they will divide and differentiate into three main subtypes of immunocompetent or mature T cells. So from a common immature T cells, they will divide themselves to many cells, and then they will differentiate themselves into three types, which are T helper cells, cytotoxic T cells, and also regulatory T cells. T helper and T regulatory cells, they will acquire surface protein called CD4, while the T cytotoxic will acquire a surface protein called CD8. So this CD4 and CD8 is the difference between the T helper Treg and also the T cytotoxics. The T cells also acquire surface proteins that function as T cell antigen receptors or TCR. For activation of cell-mediated immunity, this majority happen in lymphoid organs and tissue, where activation of T cells by specific antigen presented by the APCs. So the APCs will engulf and digest the pathogen, and then some part of the digested pathogen as an antigen will be displayed on their MHC class 2. So the antigen on the MHC class 2 will be presented by APCs to the helper T cells and helper T cell looking on the antigen using the TCR. So using TCR is how T cell will recognize the antigen presented by APCs. APCs present the antigen displayed on its MHC class 2 in the form of antigen MHC class 2 complex to the T cell receptor. So the TCR on the T cell is like a barcode scanner that will scan the antigen and recognize them. The function of T cell subsets. T helper, the main function of T helper is to secrete cytokines, activate cell-mediated and humoral immunity from this cytokine that being secreted or produced, stimulate and also amplify macrophages activities. So basically, T helper responsible for the whole immune response, from the activity of innate immunity in the form of macrophages activities to the adaptive immune response. Second, the T cytotoxic T cells kill abnormal cells and also foreign cells. These foreign cells can be observed in the cases of organ transplantation, where the recipient will recognize organ from the donor as foreign cells. Only kill cells with specific antigens that match their T cell receptors. Third, we have T regulatory cells, which control the immune response, and they basically downregulate induction and proliferation of affected T cells. They activated other T cells such as T helper and T cytotoxics to suppress other immune cells from going berserk, such as in the cases of hyperinflammation or autoimmunity, where they are attacking their own cells. So it's like to counter the activity of T helper cells, where T helper will stimulate everything, then will be down regulated, will be reduced to normal by the Treg. And we have memory T cells, which will remain for a long time in the body after an infection has eliminated, and upon re-exposure to the specific invading antigen, the memory T cells will quickly converted into large numbers of active T cells, which will provide a rapid response to the past infection. So let's have a look on the function of T cytotoxic cells or T cells. T cytotoxics provide cell-mediated response directly to kill the specific pathogen, abnormal or foreign cells. So their main function, the TC main function is to kill cells. This type of immune response is especially effective against the following types of organism and cells, such as intracellular pathogens, such as pathogens or microbes that infect our body by getting into the cell, such as viruses, fungi, protozoa, some type of bacteria, and etc. that are attacking and hiding inside the body cells. Second, the mutated cells, tumor cell, which also termed as cancer cells, and cells of tissue transplant, which is foreign cells. The activation of TC first start with the antigen recognition, where antigen recognition refers to the binding of an antigen to a TCR on the TC. So all T cells, including the TC, has their own TCR here. So the TCR will recognize the antigen being presented by the infected or abnormal body cells. We can observe here the antigen attached to the infected body cell MHC class 1, and like a lock and key system, they will bind to the TCR of the inactive cytotoxic T cell, which later will activate them upon binding. When an inactive T cell encounters its antigen complex with MHC class 1 proteins on the surface of a virus infected or cancer cells, it binds to recognize the complex. So now the inactive TC already recognize the antigen. So they recognize the endogenous or tumor antigens. Upon recognition, they will become activated and begin to proliferate and differentiate. Activated TC enlarge and divide, forming a clone, a population of identical cells of TC. A lot of clones are being produced. The activated T cells are carried by the blood from the lymph nodes or spleen to all over the body. When they meet the abnormal cells that displayed the antigens complex that they recognized earlier, they will bind to it and then kill the cell by releasing damaging cytotoxic proteins. You can observe here the activated TC will clone itself to a large number of active cytotoxic T cells, which will attack the infected body cell, and also a portion will become memory cells. And then beside self-recognition here, the antigen recognition by the cytotoxic T cell themselves, they will be also stimulated or co-stimulated by the helper T cells. APCs process the antigen and then put the processed antigen on their MHC class 2 here, and then after presentation, the T helper will convert from being inactive to an active or activated form of T helper cells. The T helper also stimulated by interleukin 1, which is secreted by the APCs. So this interleukin 1 is a type of cytokines. So we can observe here the APCs, after processing the antigen, presenting or showing it to the helper T cells, they will also produce cytokine called as interleukin to further enhance the activation of the T cell. The activated T helper and then proliferate and secrete a variety of cytokines, including interleukin 2. So this activated will clone themselves, and then the clone will produce interleukin 2. This interleukin 2 will stimulate the antigen bound T cytotoxic. Interleukin 2 also acts as an autocrine, which is they will stimulate the interleukin 2 that being released by the T helper will stimulate the other T helper to proliferate and also secrete variety of cytokines. So this is called autocrine, self-stimulation, increasing the proliferation of T helper. So this is the whole image that can you observe later on the notes. Once activated, we will proceed with attack by the TC. TC have three killing mechanisms. First, by cytolysis, which are using perforin, which act by forming holes on the plasma membranes of target cells, causing them to explode and die. So this activated T cell will release perforin, will poke many holes on the cells, and then they will explode and die. And also pre-granzymes which induce apoptosis. Apoptosis is programmed cell death. So the cell will eventually slowly dying by themselves. Second, DNA fragmentation by lymphotoxin will kill target cells by DNA fragmentation, together with destroying the cell membranes and asking the infected cell to die or suicide. The TC also will destroy the cell DNA using lymphotoxin. And second, by phagocytosis, by using the help of phagocytes, a cytokine called gamma interferon enhance the phagocytic activity of macrophages, will ingest and kill the target cell. So we can observe here, once the activated cytotoxic T cell start to attack and destroy the infected cells, the phagocyte will come to aid by engulfing or eating all the pathogens or microbe nearby. They will be released by the destroyed cells, and the phagocyte also will help in destroying the cell by eating the membrane of the infected cells. So we have two types of antigen I mentioned earlier, endogenous and also tumor antigen, which is the two types of antigen that can activate T cytotoxic cell. Endogenous antigen such as the viral nucleic acid alter the DNA of the host cell, causing it to produce viral proteins which used to produce new viruses inside the infected cells. The infected cell will displace some of the viral proteins on its plasma membrane by attaching it to MSC class 1. So this MSC class 1 together with the viral proteins will be recognized by the TC. As for the tumor antigens, cancer cell result from genetic changes induced by viruses, chemical or radiation will genetically alter the cancer cell to produce abnormal proteins which are not normally found on normal body cells. So these abnormal proteins will be attached to the MSC class 1 by the cell and then being recognized by the TC as abnormal, and the process of destroying it will activate it.